Hey guys, we're starting a new unit today and we are going to be focusing on enzymes. I'm going to split this video into actually two videos, which one is going to be just kind of an overview of enzymes and what they are. And the second one is going to be different factors that affect enzymes and um, stuff like that. So it's going to be two videos, but they go back to back. Okay. So we're going to start out by talking about just what enzymes are. We're going to kind of like transport ourselves mentally back to the first unit, which was about biomolecules, because enzymes are this unit that we're starting right now, unit three, is about energy and how cells use energy and how they create it. So the reason that we're starting with enzymes for that is that enzymes are biological catalysts, and catalysts mean that they speed up reactions that are happening in your body. So, actually, without enzymes, they're responsible for doing like a ton of digestion, but also for like building up things in your body, building muscle, building more proteins, building more DNA. So if you didn't have enzymes, your body wouldn't be able to actually do those processes fast enough for you to survive. So they're super, super important. And the way that they speed up reactions is actually by lowering the amount of energy needed to start the reaction. And that's called the activation energy. You might have learned in middle school about different chemical reactions, and that is what your body's doing all the time and what enzymes are actually working on is making these chemical reactions easier for your body to do by reducing the amount of energy that's actually needed to make them happen. Okay, so I wanted to give you guys some examples of enzymes, and you're going to be doing a project this week that lets you kind of focus in on one of them. But some examples, these first ones, amylase, lactase, and sucrase, are all enzymes that break down different sugars. They break down carbs. Protease is an enzyme that breaks down protein. Oops, not P. Down protein. And then lipase breaks down fats, breaks down lipids. And you might be noticing a trend here, which is that all of these words end in ace. And that is true that enzymes typically their names will end with the letters ASE. I'll just put that up here. So names of enzymes end in ASE. And names of sugars, you might remember from our carb discussion, end with ose. And that will also be something you'll see in our lab this week, that lactase breaks down lactose. Tose is the sugar and tase is the enzyme. Okay, back to our examples. DNA polymerase is the last one on this list, and that is something that actually creates polymer, which hopefully you remember that word from the first unit, out of DNA. So this builds new DNA strands so that you can actually make new cells. So here I've listed some different types of enzymes. The first three types are digestive, so they're going to be responsible for actually breaking down food that you ate. And then DNA polymerase is a synthesis enzyme, so it is responsible for actually like building up something new. When you put together digestion and synthesis, that equals your metabolism. Your metabolism is all of the chemical reactions happening in your body. So, a lot of people think about metabolism as just being like how fast your food breaks down, but it also has to do with how fast you're able to rebuild things back up, whether that's a protein or DNA or new membrane, whatever it is. That's also part of your metabolism, which of course is related to energy, which is the topic of this unit. Okay, so now let's look at a model of an enzyme reaction. So I'm going to scroll down here and I've drawn these shapes that represent an enzyme reaction occurring. So it happens in these three steps, and each of the arrows kind of shows like the transition to the next step. So I'm going to start over here because this is the beginning. Just to make this a little bit easier, I made my enzyme E-shaped for enzyme. So this is the enzyme. The enzyme is always a protein. So we talked about when we talked about proteins, how the shape of them is super, super, super important for their ability to function. And that's why we spent so much time kind of talking about protein folding and all that stuff. Because for enzymes, if the shape of the enzyme isn't perfect, it can't match the shape of the other molecule, which is called the substrate. That's this I shaped molecule. The substrate doesn't have to be a protein. So this could be any type of molecule, and it will be modified by the enzyme. So it's really important that the enzyme is the proper shape, and the only way that it will be the proper shape is by folding correctly through the protein folding process. And we'll talk more about the protein process again when we get to DNA a little bit later on. So that's step one, is just there is an enzyme, there is a substrate. Then what happens is the enzyme and the substrate come together and attach. So here's, if the enzyme's the wrong shape, the substrate can't come together with the enzyme. The line that is highlighted in yellow is called the active site. And this is the location on the enzyme where the substrate attaches. So the most important thing to be the right shape is that active site. When the enzyme and the substrate are combined together, it's called the enzyme substrate complex. Which is just a fancy way to say that they're attached to each other. This is, I'll just say enzyme and substrate attach. And during this time when the complex is formed is when the enzyme is actually doing like the modification of the substrate. So what happens after they detach is something like this. This, in this case, it's a digestive enzyme because it broke the substrate up. So now instead of a substrate, you have products, products. Oops. And this is what obviously is produced. So I'll just say result of the reaction. And then, of course, you still have your enzyme, which this is an important fact, is that the enzyme was not changed by the reaction. We'll say was not modified by reaction. So obviously, when you look at different enzymes, they're not always going to be E-shaped. The way that you'll be able to tell, like, oh, that is definitely the enzyme is by looking at, is it the same as it was at the beginning? And if it's the same as it was at the beginning, it's the enzyme. And if it's different, it was the substrate and it's now the product or product. Okay, so that's kind of the reaction in pictures. And then I'm going to talk about two important words that apply to all enzymes, and they are reusable and specific. So, reusable obviously means it can be used again, but for our purposes, what it really means is that the enzyme is not altered or damaged from the reaction. So, it can be used over and over. Specific means that the enzyme will only fit with one substrate. So each enzyme only matches with one specific substrate. And you can think about this sort of like, say, like a lock and key. So one specific lock has one specific key. And that's how it is with enzymes too. So lactase can only modify lactose. It can't modify sucrose. It only can work on lactose. And we're going to see that in our lab too this week. Okay, that's it for part one. So there you go. And then make sure you also watch part two, which is coming up next. Bye.